Okay, we will make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at cell fractionation. Okay, so last lesson we looked at microscopy. Today we're going to be looking at the second part of the specification, which is cell fractionation. We're going to start off by looking at cell fractionation, then we're going to zoom in into ultracentrifugation, and then we're going to finish off with two exam questions that I've picked out, which are extended mock questions. Normally, when this comes up in an exam, you will normally be given sort of extended answer questions, so 4, 5, or 6 markers in an exam, and they normally come towards the end of an A level AQA biology paper 1, where you have sort of 15 marks dedicated to extended exam practice. So you have a 5 marker, a 4 marker, a 5 marker, and a 6 marker at the end of paper 1. And normally one of the questions is on cell fractionation and ultracentrifugation. Okay, so it is worth a lot of marks. That's why it's a really important topic to focus on. In terms of the specification, we looked at the first part of the specification in our last lesson. We looked at microscopy in our last lesson, and we looked at the difference between transmission electron microscopes and scanning electron microscopes. We also looked at the formula for magnification, which is the size of the image divided by the size of the real object. Today, what we're going to be focusing on is this part of the spec, the principles of cell fractionation and ultracentrifugation as used to separate cell components. Okay, so to start off with, we're going to look at what cell fractionation actually is. Okay, so cell fractionation is used to break down or open cells so you can isolate organelles and look at their structure and function. Okay, so imagine this. So we know that a lot of sort of organisms have cells. Multicellular organisms have multiple number of cells. Okay, so we've looked at plant cells, we've looked at human cells. Okay, now if we wanted to look at each organelle in detail, how do we actually look at the organelle in more detail? How do we isolate the organelle and look at the organelle in specific in more detail? How does that actually happen? Well, that happens through cell fractionation. Cell fractionation will get your sample. Let's say we've got some tissue or something, human tissue or so, anything like that, or plant tissue. We've got that plant tissue. What it does is that it breaks open. First of all, it breaks the plant tissue into smaller and smaller pieces. And then what it does is that it breaks the individual cells in the plant tissue. So it separates out into its plant cells. So you have different plant cells. Then it will break the cells. It will break the plant cells. And if the plant cell breaks, what's going to happen? It's going to release all of the organelles that are within the plant cell. Okay, so for example, we have the nucleus. The nucleus would come out. We have the mitochondria. The mitochondria will come out. We have the ribosomes. The ribosomes will come out. Okay, so all the internal organelles that are within the nucleus, they will sort of burst out of the nucleus because we have broken open the cell. Now, once the organelles have come out of the cell, we can look at each organelle in much more detail. We can extract each of the organelles, the specific organelle, and we can have a look at it under a microscope in much more detail. And that's the purpose of cell fractionation. It is literally getting tissues, plant tissue, human tissue. It is literally breaking it down into its individual cells and then breaking the cells down so the organelles can be released in solution. Once the organelles are released, then we can analyze specific organelles that we want to. For example, the nucleus. If we want to analyze the structure of the nucleus, we can use ultracentrifugation to extract the nucleus, and then we can examine the nucleus under a microscope. Okay, but for now, if we stick to the definition, we're going to be looking at all of the details today. It's cell fractionation is just a technique which is used to break open cells. So the organelles which are released, you can isolate them and you can look at their structure and the function that they perform. Okay, now what we're going to focus on today is basically how we can prepare this sort of apparatus, basically how we can actually analyze um, our structures and how we can get it so the structures are by themselves with nothing else. Okay, so the cells firstly are prepared in a solution which must be cold, isotonic, and buffered. Okay, so basically we have our tissue. Okay, let's say we've got onion tissue, for example. Okay, we've literally cut out the onion tissue. We've used a knife and we've cut out the onion tissue. We have a layer of onion tissue. We have pieces of onion tissue. So what we're going to do is we're going to firstly cut this onion tissue into small, small pieces. Okay, so we have small, small pieces of onion tissue. Okay, and then what we're going to do is obviously we want to break those small pieces down into even smaller pieces. And one way that we can do this is by mixing it with a solution. If we mix it with a liquid, it will dissolve in the liquid. Okay, and if it dissolves, it will become smaller and smaller. Okay, because we want to analyze organelles. We want the cells to be broken. We don't want the tissue to be broken. We're going to break that anyway. We want the individual cells to be broken. And in order for those cells to break, we need to mix the tissue that we have cut up, the tissue that we have broken down into small pieces. We need to mix that with a solution. And that solution must be cold, isotonic, and buffered. Okay, and we're going to have a look at why it needs to be cold, isotonic, and buffered. Okay, so in terms of the solution, so we've cut up our pieces of onion tissue into small pieces and we've just put it into a solution. Now that solution, number one, has to be cold. The reason why it needs to be cold is because the enzyme activity needs to be reduced. Okay, because ultimately we are analyzing the organelles. We don't want to cause any damage at all to the organelles. Okay, we don't want to cause harm to the organelles. We want to observe the organelles. If we cause damage to the organelles, then we're not going to be able to see the organelles structure. There might be some damage done to the structure. It also might not allow the organelle to carry out the function that it normally does. So first of all, the solution must be cold. It must be cold to reduce enzyme activity. Okay, it will reduce the enzyme activity so the enzymes aren't able to cause damage to the organelles. Okay, because this will affect how the organelles look and how they function. Okay, so that's the key point there. So first point, it needs to be cold. Okay, so the solution that we cut up the tissue and put in, it has to be cold. The second thing is that it must be isotonic. Okay, now isotonic you've probably seen before. Isotonic means the same water potential. That's what isotonic means. Hypertonic means that there is too many solutes, so a lower water potential, and hypotonic means the water potential is high. Okay, there are less solutes and the water potential is high. So isotonic, the organelles must be in the same water potential as the solution. Okay, so this solution that we mix it in, it must have the same water potential as the organelles. Okay, because if you imagine, if I drew an image, yeah, and these are our organelles so we've got our organelles on this side, and inside the organelles, we've got this many water particles. Okay, and then in the solution we put in very dilute concentration of water. Okay, and that means that there is a lot of water particles. Okay, like this. So what's going to happen is that these water particles are going to move. They're going to move into the organelle by osmosis. Okay, so the water particles are going to move by osmosis across a partially permeable membrane into the organelles. And when they move into the organelles, what's going to happen is that these organelles can burst, basically. Okay, it can cause damage to organelles. Organelles can burst. And if they burst, we're not able to see the function that we want to. We can't observe the structure in much detail. Okay, so and this is the same vice versa. If the solution that we use is hypertonic and it has a very low concentration of water, what can happen is that the water can move out from the organelles into the solution, and that can cause the organelles to shrivel. Okay, and again, that won't allow us to see the structure that we want to see. So the organelles must be placed in an isotonic solution. Reason why is because we don't want the organelles to be damaged. Again, it's all because we want to prevent the damage to the organelles. We don't want the organelles to shrivel or burst. If water moves into the organelles because of a high water potential of solution, then the organelles will burst. If the solution that we use has a high concentration of solutes and a low water potential, then water will move out of the organelles and it will cause the organelles to shrivel. Okay, and again, that will affect the structure of the organelles, which we don't want. So we want to prevent osmosis from occurring as much as we can because we want to keep the normal structure of the organelle so we can observe it as it is. So it needs to be isotonic, which means that it must be in the same water potential as the solution. Because if it's not, then it can cause damage to the organelles. It will cause the organelles to shrivel or burst. And the final point that you need to know, the solution should also be buffered. And the reason why it must be buffered is to prevent damage to the organelles. Again, once again, all the reasons of this, it needs to be a cold, isotonic, and buffered solution is to prevent damage to the organelles. Okay, we don't want to damage the organelles at all. We want to observe the structure of the organelles and the function of organelles. We don't want our experiment to affect how the organelle works or acts in a different surrounding. Okay, so it needs to be buffered. So again, we don't want the conditions to be too acidic, too alkaline. We don't want to denature any part of the organelle. We don't want to affect any, we don't want the pH to affect any of the function. So it's a control that we need to have in order to get the organelle as it is, carrying out the function that it normally does. Okay, so the three factors of the solution that we need, they need to be cold, isotonic, and buffered. Okay, those are the three factors. Okay, so as I said on the previous slide over here, we're looking at cell fractionation. And in cell fractionation, what we are doing is we are breaking open those cells, the tissues, the cells, and the cells will release the organelles into the solution, and then we can extract each of the organelles and we can observe the organelles in much more detail. What does it do? How does it look? And what the function of the organelle is. The first step to that is we need to break open the cell. Okay, and how we do that, we first of all, if we have a large piece of like plant tissue or something, we use a knife to cut it up into really small pieces. Okay, so we use a knife to cut it up into really small pieces. And once you've got it into really small pieces, you can mix it into a solution, because when you mix it into a solution, it will make the pieces even smaller. It will dissolve some aspects of the cells, and that will cause the cells to become even smaller and perhaps even release their organelles. Okay, and the conditions for that solution, it needs to be cold, isotonic, and buffered. All three need to be present in order to make sure that you don't cause any damage to the organelles, because we don't want to affect the structure of the organelles. Okay, so there are two main steps for cell fractionation. The first step is homogenization, and the second step is ultracentrifugation. Okay, and I've already spoken over some of these aspects, but the following slides will now explain the sort of method that you would go through cell fractionation, how you can sort of obtain a solution where you can observe the organelles under a microscope and look at their function and structure in more detail. The two steps, the main steps that we are going to go through, are homogenization and ultracentrifugation. Okay, so we're going to start off by looking at homogenization. Okay, now homogenization is basically the part when you break open a cell. Okay, that's what homogenization means. Homogenization means to break up. Okay, that's what homogenization means, to break up. So we know that the cell must be broken open. Okay, and normally what this is done is this is basically done using a blender. Okay, so let's say we had some tissue. Okay, we had this layer of tissue like this, for example. So this is our tissue sample. Okay, and we'll just say that this is plant tissue. Okay, so this is our plant tissue. So what we're going to do is we're going to cut up this plant tissue, this tissue sample that we have. This plant tissue can be taken from anywhere. It can be taken from a plant. You can just cut up a plant and obtain a plant tissue. It could be taken from a leaf. It could be taken sort of anything like that, basically. What we're going to do is this tissue sample, we're going to cut up, first of all, using a knife. Okay, so we're gonna cut it up into small pieces. Okay, as small as we can get with a knife. We're not gonna get it this small. I've just drawn this as an example. Okay, but in reality, it's gonna be a bit bigger than this. Okay, so we've cut up the tissue sample into small pieces. Okay, now once we've cut it up into smaller pieces, what we're gonna do is we are going to put this into a beaker. Okay, so this is our beaker, and this is our cold, isotonic, and buffered solution. So this is our cold, isotonic, and buffered solution. We're going to add the pieces that we've cut up into this cold, isotonic, buffered solution. Okay, and we're just going to give it a stir. We're then going to add this into a blender. Okay, so once we've added this, we're going to blend it. Okay, so we're going to add it into a blender. Okay, and we're going to turn the blender on. We're going to leave it for a minute, okay, and allow it to blend. And once it's been a minute, we're going to pour it out and we're going to filter the solution. Okay, so we're going to pour it out. So after a minute, filter the solution into a beaker. Okay, and that's to remove any large sort of pieces that are still there after blending it. Okay, so we don't want any large pieces to be present. We basically just want dissolved solution to be present. Okay, so we had our tissue sample, we've cut it up into smaller pieces, we've put it into our cold isotonic and buffered solution, we've stirred it, and then we've put it in a blender for a minute, and then we've filtered the solution that comes out from the blender. And ultimately, what you will have by the end of this is a beaker that basically has your solution and sort of any dissolved um, tissue that we originally had. You wouldn't be able to see it because we filtered it, but you will still have the dissolved pieces there. They're just too small to see. Okay, and that basically tells you that you have broken open your cells and the organelles are there in the beaker now. Okay, because they have dissolved within the solution. They're there, you've just broken up the cells. Okay, so the reason why we filter it is to remove any large debris. Okay, so that's all of homogenization. Homogenization means to break up. And all we are doing is we are getting that tissue sample, we're cutting it up, pouring it to a cold isotonic and buffered solution, we're blending it, and then we're filtering our solution after it's been blended into a beaker, and that's to remove any large debris that we had. The organelles are now in the beaker, and now we can work on looking and analyzing them individually. Okay. So the next step, the next stage, we've looked at the first stage, which is homogenization, and that's all you need to know about homogenization. In terms of centrifugation, okay, now we've got at the moment what we have is a beaker with our organelles, which are just present in the solution. Okay, now there's going to be millions of organelles in there. Okay, you're going to have nuclei in there, you're going to have chloroplasts in there, you're going to have mitochondria in there, you're going to have lysosomes in there, ribosomes, you're going to have loads of organelles in here. Okay, so you're going to have nuclei, you're going to have mitochondria, you're going to have ribosomes, and loads more organelles. Okay, um, there's loads of organelles that you're going to have in that solution. What was our aim? Our aim was to extract specific organelles and look and study their structure and function in more detail. At the moment, we're part of the way through there. We have a solution containing a lot of organelles. Okay, but how do we actually extract a specific organelle and study that organelle in detail? And that's why centrifugation is there. Centrifugation will separate the organelles based on density. Okay, so the centrifugation will separate the organelles based on density. So the filtered solution, which we have just filtered, as you saw on this previous slide, we've got a solution that just has organelles in it. The filtered solution is then spun at in a high-speed centrifuge, and this will separate the organelles according to their density. So again, we want to obtain specific organelles. We don't want a bunch of random organelles. We won't be able to analyze them ever. Okay, we want specific organelles so we can analyze each one in detail. Okay, so that's why we centrifuge. Centrifuge is a sort of machine where you put sort of test tubes in, and then it will literally spin the test tubes at sort of high speeds. And the faster it spins the test tubes, the more sort of organelles will sink to the bottom. Okay, now different organelles have different densities. That's why we use centrifugation. We know that it separates the organelles based on density, and that's why some organelles are heavier than the others, some are lighter than others. Okay, so organelles have different densities. Okay, so for example, the nucleus is the heaviest organelle that you have in a cell. Okay, so if you centrifuge a solution, the nucleus is going to sink to the bottom first because the nucleus is the heaviest. Okay, it's going to sink to the bottom first so you can extract the nucleus. Okay, so the centrifuge spins at high speeds, and the centrifugal forces cause the pellets to form at the bottom of the tube. Okay, and the pellet is just the thing, the solid part, which moves to the bottom, and it's made up of the densest organelle. So for example, if you've put this solution into a test tube like that, for example, at the start you won't see anything. It will be a clear solution. After you centrifuge, you will get a test tube which has the same solution, but you will see sort of darkness at the bottom. Okay, and that means that this is the organelle which is the densest. This is the densest organelle that you have at the end of the first centrifugation. Okay, and then what you can do is you can remove that organelle. You can, this thing, that this colorless part, you can just remove it into another test tube, and you can be left with the test tube that has the organelle in it by itself. Okay, and in this case, this will be a nucleus. And then the empty test tube that you have, where you've poured this white thing out into, you can put that back into the centrifuge, and you can centrifuge it at an increasingly faster speed. Okay, if you centrifuge at an increasingly faster speed, it will remove another organelle. Okay, so the process is repeated at increasingly faster speeds, removing the supernatant, which is the liquid, each time, leaving behind the pellet. Okay, and the pellet is the organelle that we are talking about. So if we have a look at an example, initially, this is our solution. Okay, this is our solution after we've blended it and we've filtered the large debris out. That's our solution. Okay, now we centrifuge. Once we've centrifuged, we will get a test tube that has the same solution, but you will have a dark part at the bottom. Okay, and this is your densest organelle or heaviest organelle. Okay, so that's your densest organelle at the bottom. What you can do is you can pour this liquid thing that you have at the top is called the supernatant, and this solid thing that you have at the bottom is called the pellet. Okay, so you can remove the supernatant out into another test tube. Okay, so if you just pour it out into another test tube, the supernatant, this is the supernatant that was left, you will be left with the dense organelle at the bottom in the original test tube that you have. And then you can analyze this organelle that you have left. Okay, then this supernatant that you removed over here, you can centrifuge this again. Okay, you can centrifuge again at a faster speed than you did originally. Okay, centrifuge at a faster speed. If you centrifuge at a faster speed, the next thing that you're going to get is you're going to get a test tube that still has the same supernatant, but you will have a different organelle which sinks to the bottom because you've centrifuged at a faster speed. And this will be the second densest organelle. And then you can do the same process. You can separate out the organelle, you can pour the supernatant into another test tube, and then centrifuge again at a faster speed. Okay, and that will separate your organelles according to density, basically. Okay, and that's basically centrifugation. Okay, and there's an order in which the organelles go in, the density of organelles, and you need to be aware of the order. Okay, so the order of cell fractionation is as follows. Okay, so you have the nuclei. Okay, that's the most dense. Okay, so over here, when we centrifuge first, the densest organelle that we obtain is the nuclei. Okay, that's the heaviest in a cell. And then the second heaviest is the chloroplast. Okay, so over here, the second densest organelle that we're going to have is the chloroplast. So each time you centrifuge at increasingly faster speeds, you will receive a new organelle until all the liquid is finished, basically. So you're going to have the nuclei first, that's the most dense. Then if you centrifuge another time, you're going to have chloroplasts. Then if you centrifuge another time, you're going to have mitochondria. Then if you centrifuge another time, you're going to have lysosomes. Then you're going to have the endoplasmic reticulum. And then you're finally going to have the ribosomes. The ribosomes are the least dense. Okay, so they're the lightest out of all the organelles which are within a cell. Okay, so the first thing that comes out is nuclear, but normally when you're given an exam question, it's normally out of the first three that you normally get asked. So I would know the whole thing. There's acronyms that you can learn to remember the order of the organelle fractionation, but know the first three especially: nuclear, chloroplast, and mitochondria. Okay, so that's the order of organelle fractionation. And that is basically everything in terms of content for cell fractionation and ultracentrifugation. If I was to quickly summarize it, first of all, you homogenize. You get your tissue, your onion tissue or wherever, and you cut it up into smaller pieces. Once you've cut it up into smaller pieces, you then place the smaller pieces into a cold isotonic and buffered solution. The reason why it needs to be cold, isotonic, and buffered is because you don't want to damage any of the organelles. Because ultimately, the purpose of us actually doing this is to observe the organelles, the structure, and their function in more detail. Okay, then what you're going to do is you're going to stir the solution with the small pieces of tissue that you have cut apart, and then you're going to place it into a blender for about a minute, and then you're going to filter the solution after you've blended it. Okay, and that will remove any large debris that you have. You're then going to put this solution into a test tube. Okay, and what you're going to do is you're going to put it into a centrifuge, which is just a machine that will spin the beaker at increasingly faster speeds. Okay, and then when you centrifuge, what you're going to have is that you're going to have the densest organelle being collected at the bottom. It's going to sink to the bottom, and you can see where it sinks. It will just be a bit darker at the bottom of the test tube. Okay, and then you can separate that. The thing that sinks to the bottom is called the pellet, and the liquid part, which still is normal colorless, is the supernatant. So each time you are collecting the pellet, the thing that has sunk to the bottom, and then you are removing the supernatant and putting it back into the centrifuge so you can centrifuge it at a faster speed to collect another organelle. And remember, centrifugation is all due to density. It will separate the organelles based on density. And you need to know the order of the organelle fractionation. The most dense is the nuclei, then it's the chloroplast, then it's the mitochondria, then the lysosomes, then the endoplasmic reticulum, and finally the ribosomes.